I'm sorry, but we are very short in time, and we don't want to do any compensation on science and lectures, so the only thing we can do is drink coffee faster, and maybe we will need to eat a little faster later. So we are moving to the second health session, and I'm very proud and very privileged to invite Professor Keren Avraham, which is the Vice Dean of the Medical School at Tel Aviv University, and the rest you'll see by yourself.
Okay, it's such a pleasure to be here. I do apologize to all of you for not being here yesterday. We had a faculty event, of course, that I wanted to attend. So I got in a taxi at 6:30 this morning, a few hours of work, and here I am. And it's just incredible because every time I lifted my head, I realized I've come to such a different climate, such a different view, and it really is stunning here. So I'd like to introduce this session, and I will do so by inviting our first speaker, who's Professor Dan Pe'er, who is a good friend and a colleague. And it's such an honor for us to be able to hear him. He comes, as you all know, from Tel Aviv University, from the Faculty of Life Sciences. And I'd have to say that really close your ears a little bit. It's my expertise, so I can say that. That Dan really is a pioneer in targeting of therapeutics, going from molecules to immune cells. And years ago, he had a review in Nature Nanotechnology, which is still the most cited review in the area of nanomedicine. He then had a real landmark science paper, many papers, many patents in between. In fact, he has about 110 pending and granted patents. And just in 2018, just last year, so you can see that he continues this really remarkable trend, he published the first system in delivering mRNA, specific delivery of mRNA for IBD. So he does so much more. He also has some administrative roles. He heads now the Spark Translational Center at Tel Aviv University. He's now, I think, got a new position as Vice Dean for Research at the Faculty of Life Sciences. So there's really no end, and we're, of course, involved in multiple projects, and he's got, not products, but projects. No creams, right? No creams yet. So without further ado, please, Danny.
Okay, thank you so much. Somehow. Our lab is studying how to manipulate leukocytes' function in order to generate novel therapeutic approach to treat mainly leukocytes implicated disease. Everything is related to immune cells. If you're not convinced, you should look at a partial list. Actually, there are much more of this, but this is a partial list, and we are very much involved in all those bluish diseases, going all the way from a concept idea into potential products. So our lab is divided into two major groups, people that are doing drug delivery system, mostly lipid-based. We synthesize those lipids, and if I have time, I'm going to talk a little bit about this. We generate monoclonal antibodies, a protein engineering. Some people think we are good at this. Actually, we are not. And the other side is something more serious, which we call biology. Biology is almost more complex than technology, and technology always runs faster than biology. And so this part of the lab is a little bit more problematic, I think, over the years, but I'll hope to give you some examples in the next few minutes. Okay, in an ideal situation, we do the drug discovery together with the drug delivery. We are mostly interested in manipulating leukocytes' function through their genome or their transcriptome. So this is very challenging, as you probably know. And I'm going to give you some challenges of basically how RNA delivery and gene manipulation, leukocytes. You're a very diverse audience, so I'm not going to go into details. Most of the things that I have here have been published either recently, very recently, or a little longer. But basically, we have the challenge of any other regular challenge of efficient encapsulation when we want to inject something that will go through the bloodstream into specific cell types, we need to entrap the RNA, and we need to do it in a very efficient manner. Okay? Specificity, targeted delivery platforms, specificity is very important, and this is happening in any related delivery. It's not related to immune cells. Evading clearance mechanism, avoiding toxicity and immune activation, carrier internalization, and endosomal escape, all are very, very important. But just imagine that you're now going to penetrate into the holy grail of your immune system. And you need to do it very quietly. And what we're trying to do is to penetrate into very unique cells of the immune system, T cells, B cells, mononuclear phagocytes, subsets of T cells, subsets of B cells, and manipulate them. The hardest one, and if you want to get probably a Nobel Prize, will be memory B cells. So if you will be able to manipulate memory B cells, you're most likely going to impact health. And the reason is nobody can do it. So viruses can penetrate, actually, quite efficiently, but then those tiny amounts of memory B cells will die, and they are dying very fast, within 20 hours, less than 24 hours. So somehow they identify us. And we are using nanotechnology and basically very high throughput technology in this field. And this is very advanced already. We have the first product that was approved by the FDA in August 10 last year, which is for hereditary TTR, amyloidosis, rare disease by Alnylam Pharmaceutical. And this is siRNA that silences genes in the liver with lipid nanoparticles. And this is the first example, and there are a few now phase 3 trials on the way, and quite a lot of phase 2 and phase 1. I'm going to start by telling you a few stories, again, diverse audience. The first one will be on inflammatory bowel disease. Crohn's, colitis, ulcerative colitis, and chronic pouchitis are the known IBD, and the most advanced one, or most known one are Crohn's disease and ulcerative colitis. And we tried to understand quite a few years ago why cyclin D1, a gene that is responsible for cell cycle regulating, is actually upregulated in immune cells during inflammatory bowel diseases. And we set up a strategy to target gut mononuclear leukocytes and selectively silence this cyclin D1. And the reason was that there are a few papers demonstrating that there is a connection between proliferation and migration. Okay? So this is a bit bizarre, okay? That immune cells that proliferate also migrate better to the gut. So the idea was if we stop the proliferation, maybe we'll stop the migration. One of the problems in IBD is the migration of immune cells into the gut that destroy the tissue. So we set up a very naive strategy, but we discovered something very interesting in this strategy. So first we had to create an antibody that target immune cells that home to the gut. And so we created a few clones for this, and this is the anti-beta 7 antibody, okay? And we have shown and demonstrated that actually we can silence gene very efficiently under shear flow condition in vitro, and we can do it also when we have a beta 7 knockout mouse, and that was done. We also found a new role for cyclin D1 as a co-transcription factor that controls some of the pro-inflammatory cytokines. And this had a lots of important things. It's actually part of the molecular biology of the cells. It's a textbook already. From the 6th edition of Alberts and so on. And so we discovered that cyclin D1 is what we call a colboinic. It can go in and out the nucleus and do a lots of things, including silencing some of the pro-inflammatory cytokines during IBD, but also in rheumatoid arthritis. So what happened with this? So you publish a fancy paper, but then you want to do something else with this. And what we wanted to do is to establish a company. So I'm a translational guy, and I've been like this for my PhD at Tel Aviv University. So we basically created a spin-off company somewhere in the mid of 2007 that is called Leukobioscience. It's a virtual company. What helps us a lot was this paper was published in February 2008. And the same week we were on the cover of Business Week. So this is good in Business Week. You see I had more hair. And the thing was that we had to do something with this, and we talked to many companies. One of them is a small company called Genentech, part of the Roche group. And Genentech said, we don't understand what is this creature, but we do understand how to make monoclonal antibodies. So what happened is, and I'm telling you a very long story short, what happened is that they developed an antibody based on this that is called etrolizumab that was recently approved. It still have 4 phase 3 clinical trials worldwide, and it's very interesting. And I made a career based on this antibody, which is actually fun. Okay? And the beauty of this was that the competitor antibody that is marketed by Takeda from 2012 was generated in the same lab. Okay? So people can, this is a hype, and people can use the hype. Okay, and we know exactly where it's mapping, and I'll tell you why it is important. But remember cyclin D1, because I'm going to talk about another disease called mantle cell lymphoma. It's quite of a rare disease. It's an aggressive form of a B-cell non-Hodgkin's lymphoma, about 6% of the non-Hodgkin's lymphoma. In Israel, for example, we have about 13 to 15 patients. And the median survival now, it's actually improving. It was 2 to 3 years, now it's 5 to 7 years. It's one of the most poorest among the B-cell malignancies. But it has a very unique genetic hallmark. So in 80% or 85% of the patient, there is a translocation. The gene that encodes for cyclin D1, this protein, the cell cycle regulator, juxtaposed before a very strong promoter and start to produce cyclin D1 in B cells that do not proliferate via cyclin D1. So basically, most of our B cells are proliferating through D2 and D3, other cyclin D proteins. And that's very interesting. So basically, or in principle, we have a personalized medicine approach here. We can target those patients, and we can develop a drug based on this. And what happened is that together with Mark DeLay, then in the Broad Institute, now in Karolinska Institute, we sequenced more than 80 patients and revealed 2 transcripts. One is short and one is long. And together with Alnylam Pharmaceuticals in Boston, we screened. When I say we, is this nice lady, Dr. Shiri Weinstein, then a PhD, now an MD PhD, was in my lab, now an ophthalmologist in Sheba Medical Center. And she screened 500 sequences by electroporation. This is another way to actually kick people from your lab, give them the opportunity to screen in cell lines a lot of sequences. And we actually identified one that was very, very potent. And this potent sequence was in the range of picomolar concentration, and we decided to move and take it forward. The problem was that we could not publish it, because our title was mantle cell lymphoma is a therapeutic target for, cyclin D1 is a therapeutic target for mantle cell lymphoma. And about 3 years before that, there was a paper in leukemia with the opposite title. Cyclin D1 is not a therapeutic target for mantle cell lymphoma, and now try to publish it. So we tried, it took us about 2 years. Nobody wanted to publish it. Then Shiri decided she needs to leave, and she needs to publish it. So she found this journal, PLOS ONE, and she said they accept everything that is scientifically sound. So we submitted, and we got 2 reviews. I'm getting this a very long, short story, which was long. One of them said, it's very interesting, you should revise it, there is some work to do. And the other one said, I think it should be rejected, based on the fact that it's known that cyclin D1 is not a therapeutic target. There was a paper in leukemia, and that paper was cited 200 times. And that means it's correct. So I have to do a stop and to tell you about this paper. This paper was cited more than 6,000 times, that Karen mentioned. Okay, and ISI recognized it as the highest cited paper in the field of nanotechnology for medical application. The problem was that it's not correct. Because in 2007, we knew something that is called EPR, and we generalized it. So, just as a rule of thumb, it doesn't mean if it's highly cited that it's correct. So we had to revise it. And we recently was approached about a year ago from a Nature journal, Nature Communication, and asked us to write a basically progress report of this field. And it came up to be very depressing, But it has a good ending, I think, because we actually learn what are the factors that we need to change over the time and what we have learned from clinical failures. So I'm coming back to the story. And then, when we had a very good sequence, and eventually this was published in PLOS ONE, a little bit of pressure always help. So Itai Antoine Tucker actually joined Shiri, and I gave them another goal to do it systemically. So use this delivery system or a modified version and inject it into mice, and let's see if it works. The only problem was that we learned, it's very easy to be a PI, right? You just say, that's great, do it, but in fact there was no animal model. So it took them another 2 years to calibrate a model. And Shiri is like a gorilla, okay? She is very serious in what she is doing. She pushed, and Itai was a cup, basically. And you can see that eventually, you see the admiration of the little guy to the mentor. So we had to develop a model, and we developed a model that is now available in Charles River and actually also in Jackson laboratories and was published. And with this model, we show that actually there is a very good engraftment in cyclin D1, in mantle cell lymphoma, also from patient samples. And you can actually see the model here. And you can knock down, whatever. So I'm not going to go into the details because I see I don't have time. But I'm going to tell you that it has good ending, and we basically started to use microfluidics, and we had a streamlined process. We knocked down this, and it seems to be working very nicely. These are some of the data, and we did all the assays in competition, et cetera, and in vivo blinded experiment in Charles River that ended like this, okay? And this was published. So basically, we inject, this is a targeting vehicle for us, an anti-CD38 antibody that was available by Sanofi. And what you can see here is very nice change in the survival curve, okay? However, as you can see, all the animals are dead. So a single gene is not important, although it's an initiator gene. Of course, there is secondary mutation that happens all the time, and this is a very aggressive model. So we are working on how to improve this with combination therapy, and if I have time next year, I'm going to show you. So in the meantime, we are very much interested in understand what this molecule, it's a lipid, an ionizable lipid, a pH-sensitive lipid that was approved by the FDA in Alnylam pharmaceutical formulation, and we decided to take it and basically try to do the chemistry, and behind us is Srinivas Ramichetti. He's been in my lab more than 5 years, a senior postdoc, and basically he changed the head groups, the linkers, and the hydrophobic tail. We came up with 60 lipids family. That means about 3,000 different molecules. And over the past 3.5 years, we have been screening them in different assays and in different collaborations. Recently, 2 of those lipids have been licensed and are in clinical development in BioNTech. It's a German company from Mainz that is doing personalized medicine where N equals 1, basically sequencing melanoma and breast cancer patients, learn from the mutation, see the neoantigens that are appearing, and then design messenger RNA, and then wrap it with our lipids, with our particles, and getting back to the patient within 4 weeks. And this is really an immuno-oncology approach, which we hope have been already 125 patients, according to last week data. Each one is by itself a clinical trial, but it's very interesting. Another company that licensed other lipids are Art Bioscience that develop a therapy for Duchenne muscular dystrophy. Okay, I don't have time, but we do everything on microfluidics. At the scale of a lab at the university, we can do up to 8 liters. 8 liters sterile, okay? So this is good for phase 2 trials, extensive. Okay, now we have a problem, and the problem is this. So we have streamlined the process, and we want to have like GPS system, like antibodies that recognize the specific cell types, but the problem is we cannot control the orientation. Chemistry has its limitations. Although some of the people in my lab are chemists, the others are physicians or biologists or engineers. And this is an engineering problem. So we decided to change this and move from chemistry to biology. So chemical conjugation has this problem. As I mentioned, you cannot control the orientation, and you can have bad FC or poor binding because FC receptors on immune cells can take up this very quickly when you inject this into the bloodstream. So we developed a linker strategy where we use biological affinity, basically developing an antibody, which is a secondary antibody, binds to the FC portion of the primary antibody. Now, if you chemically conjugate this linker, now you have a very nice orientation, because only antibodies that are oriented in the right configuration can bind. And the rest is history. So now I have to show you this. And before that, I want you to imagine you're a particle or a nanoparticle. Disregard the ears. Okay? And sometimes, in order to avoid mononuclear phagocytes, we have to do a chemical trick, which means we have to pegylate those things. And this is a pegylated version. Okay? And then the orientation is controlled basically by this linker. So these are the ladies behind the approach. Okay? Ranit Kedmi is a postdoc in NYU now, and Nofar is going to Germany very soon. And she's also the recipient of a Dan David Prize and Rappaport and many other things. She's very talented. And what they have developed is basically this linker approach. And it gives you the ability to do precise gene manipulation to specific cell type by not changing the primary antibody, just mixing it. And together with Itai Ben-Ar from Tel Aviv University, who is an extremely talented protein engineer, we developed three options. We started by creating an antibody and an IgG. We created a single chain based on this, and we found a sequence that could be lipidated very nicely, and we patented this. So we have a single chain lipidated formed in bacteria. And this guy can be incorporated into nanoparticles very easily. And okay, and this is the results. And I see Karen is already up and running. So I'm not going to show you anything anymore, but a short movie. Do I have a movie? I have. From the web. Okay. So I'm going to show you, because we did also this for messenger RNA. And then you have voice here? Good. This one, this one? Okay. Let's do YouTube. Good. I think it's under this very common name. Aha. Yeah. Okay.
In inflammatory disorders, the sick cells in our body are our own white blood cells. Only instead of defending the body, they attack it. The best way to fight the complexity of human diseases is to deliver the therapeutic payload directly to the sick cells. A promising technology for combating diseases uses special lipid nanoparticles. These nanoparticles contain RNA molecules that can change the way cells produce proteins and can modulate cells' function. The nanoparticles are supposed to deliver the RNA directly to the sick cells. The problem with today's technology is that the nanoparticles hardly reach immune cells, but rather affect other healthy cells instead. We at Pe'er Lab have developed a new platform called ASSET that can send RNA nanoparticles selectively to the correct cells. While existing methods of connecting nanoparticles to antibodies are extremely complicated, Pe'er Lab's method is a matter of simply mixing two solutions. And the nanoparticles are decorated with an ASSET linker. which then binds the antibodies. Each type of antibody knows to attach to a very specific type of cell. So when they decorate the nanoparticle, they will direct it only to the targeted cells. This method means releasing the RNA right where we want it, affecting only the right cells and saving the body from harmful side effects. Asset nanoparticles are a novel strategy to fight against inflammatory diseases and even cancer. And this time, it's personalized.
Hey.
That's it. That's not mine.
For the sake of your lunch, we will, Don, will you be here for lunch to answer questions? Or you're already on the plane. Okay, so grab him. Would you like, anybody have a question? He's still here before he runs out, please.
Yeah, my question is, your adapter, basically, is that binds to the FC? Maybe you said that, but I missed that.
Yes, binds to the FC.
Okay, yeah.
Yes, the answer was binds to the FC.
Yeah, okay. Anyone else?